Somewhere beneath the Egyptian Museum in Cairo, there are rooms most visitors never reach. Rooms where the dead have been waiting for 3,000 years with their faces still intact, their hair still visible, their features preserved well enough that you could pick them out of a crowd. For over a century, scholars argued about who these people were, where they came from, what their faces meant. The arguments were loud, politically charged, and almost entirely based on what everyone could see. Nobody thought to ask what the biology said. When they finally did, the answer was not what anyone expected. The oldest mummies in Egypt do not match any living population on Earth. Not modern Egyptians, not sub-Saharan Africans, not any group that current genetic databases can cleanly account for. What the DNA revealed was not a confirmation of any existing theory. It was something stranger and considerably more unsettling than that. The mummies did not need DNA to make an impression. That was always the problem. When archaeologists unsealed the tomb of Yuya in 1905, they found a man who had been dead for 3,000 years and still looked like someone. Not a wrapped bundle, not a skeleton, a face. His hair was blonde, almost yellow, still visible against the linen. His nose was prominent, straight, distinctly non-eroded by time. His features were so well preserved that photographs taken at the excavation look less like archaeological documentation and more like portraits. Yuya was a nobleman, the father of Queen Tiye, father-in-law to Pharaoh Amenhotep III. He was, by any measure, at the center of ancient Egyptian power, and he looked nothing like what the dominant popular image of ancient Egypt had always suggested. His wife Tiye was buried beside him. Her preservation was equally remarkable. The embalming process had held her features in place across 3 millennia. She was the grandmother of Akhenaten and the great-grandmother of Tutankhamun, 2 of the most discussed figures in all of ancient history. Her lineage ran directly through the heart of the New Kingdom. Her face survived intact. Then there was Tiye herself, one of the most powerful queens Egypt ever produced. Great royal wife of Amenhotep III, mother of Akhenaten, grandmother of the boy king whose tomb would eventually become the most famous archaeological discovery in history. Her mummified remains showed a woman of striking appearance, features that prompted debate among Egyptologists for decades about her origins, her ancestry, and what her prominence at court actually implied about the ethnic composition of Egypt's ruling class. Ramses the Great complicated the picture further. 3rd pharaoh of the 19th dynasty, regarded by most historians as the most powerful ruler of what was already the most powerful period in Egyptian history. His mummy, recovered and examined multiple times across the 20th century, showed reddish hair, a long, narrow face, and physical features that consistently prompted the same uncomfortable question that nobody in mainstream scholarship particularly wanted to answer directly. The question was not new. It had been circling Egyptian archaeology for as long as the mummies had been accessible. Egyptian art had always depicted a clear visual distinction between Egyptians and the Nubian populations to their south. The artifacts were explicit about this. Museum pieces showed prisoners with tightly curled hair and high cheekbones rendered in a specific visual shorthand that Egyptian artists used consistently to indicate foreignness. The Egyptians did not depict themselves that way. They depicted themselves differently. For a century, that visual evidence sat alongside the physical evidence of the mummies themselves and generated arguments that produced far more heat than light. The debates were loud. They were frequently political. They relied on what people could see and what they wanted to see, which are rarely the same thing. What nobody had done, for reasons that seem almost incomprehensible in retrospect, was sequence the DNA. The mummies had been excavated, photographed, analyzed, displayed, argued over, and written about in thousands of academic papers. Their faces had been reconstructed. Their diseases had been diagnosed. Their family relationships had been mapped through historical records. And for decades, nobody had checked the genetics. The failure was not laziness. That needs to be said clearly before anything else. Ancient DNA research has always been constrained by one fundamental problem that has nothing to do with scientific ambition and everything to do with chemistry. Genetic material degrades. It degrades faster in heat. It degrades faster in humidity. It degrades when exposed to ultraviolet radiation, to bacteria, to the simple passage of time in anything other than ideal preservation conditions. The places where ancient DNA survives best are cold, dry, and stable. Permafrost in Siberia, limestone caves in northern Europe, high-altitude burials in the Andes. Those are the environments that gave the ancient DNA revolution its early breakthroughs. Egypt looked like the opposite of all of that. The logic was reasonable. Daytime temperatures in Egypt regularly exceed extreme thresholds. The Nile Valley is not the Sahara, but it is not Siberia either. Humidity fluctuates. The tombs themselves, while sealed, were not climate-controlled across 3,000 years of occupation, looting, excavation, and handling. Geneticists looked at Egyptian mummies and made the same calculation they had made about Saharan remains. The material would be too degraded. The contamination risk was too high. Every person who had ever touched these mummies since excavation a century ago had potentially left their own DNA behind, layered on top of whatever remained of the original. Separating ancient signal from modern noise seemed for a long time effectively impossible. There was a second problem specific to Egypt that made the contamination issue even more acute. The mummies had not been sitting undisturbed in sealed conditions since their discovery. They had been moved, examined, unwrapped in some cases, handled by researchers, displayed in museums, and subjected to decades of study that was never conducted with ancient DNA recovery in mind. Nobody in 1905 was wearing gloves and thinking about genomic sequencing. They were doing the science available to them. The result was that by the time geneticists began seriously considering Egyptian mummies as viable subjects, the samples had a century of potential contamination baked into them. So the field moved elsewhere. Europe accumulated hundreds of ancient genomes. The Near East began building a substantial data set. Ancient DNA from the steppes of Central Asia reshaped the entire understanding of Indo-European migration. The science was moving fast, producing results that rewrote textbooks in real time. Egypt remained a blank. Not because nobody wanted the data. The questions surrounding ancient Egyptian origins were among the most discussed in all of archaeology. The demand for genetic answers was enormous, but wanting data and being able to recover it reliably are two entirely different things, and for decades the technical barriers held. What finally changed was methodology. Researchers at the Max Planck Institute developed screening processes capable of distinguishing genuine ancient DNA from modern contamination with a precision that earlier approaches could not match. They identified which tissues in mummified remains were most likely to have shielded genetic material from degradation. Bone. Teeth. The densest structures in the body, the ones least accessible to bacterial intrusion and environmental damage. They applied those methods to mummies from Abusir el-Melek, a site south of Cairo with remains spanning nearly 2,000 years of Egyptian history. And this time, the DNA was there. The study published in Nature Communications in May 2017 was not a small undertaking. 90 mummies, remains spanning 1,400 years of Egyptian history, from the New Kingdom period around 1380 BC through to the Roman era in 426 AD, collected from a single archaeological site at Abusir el-Melek, which gave the researchers something invaluable alongside the genetic material itself. Continuity. These were not samples pulled from scattered locations across a vast country. They were a coherent population from one place across an enormous stretch of time. That matters when you are trying to understand not just who people were, but whether who they were changed. The Max Planck team, led by paleogeneticist Johannes Krauss, extracted mitochondrial DNA from all 90 mummies. Mitochondrial DNA passes from mother to child and traces broad maternal lineages across generations. From 90 samples, they got 90 data points on maternal ancestry. which is a substantial data set by any standard in ancient genomics. For 3 of the mummies, conditions were good enough to recover nuclear DNA as well. Nuclear DNA is inherited from both parents and provides the kind of detailed population-level analysis that mitochondrial data alone cannot deliver. The contamination screening was exhaustive. Every fragment was tested for the chemical damage signatures that accumulate in genuinely ancient DNA over millennia. Modern DNA does not carry those signatures. Ancient DNA does. It is one of the most reliable ways to distinguish real ancient signal from the noise left behind by every researcher who handled the samples in the intervening century. The team was rigorous about this. The results that made it through screening were the results that could be trusted. What those results showed was immediate and striking. The ancient Egyptians at Abusir el-Melek shared almost no DNA with modern sub-Saharan African populations. The genetic distance between the mummies and populations from below the Sahara was substantial and consistent across the dataset. This was not a marginal finding at the edge of statistical significance. It was a clear pattern running through 1,400 years of remains from the same location. Instead, their closest genetic relatives were ancient farmers from the Levant, the region that includes present-day Israel, Jordan, and surrounding areas. People connected to the Neolithic agricultural expansion that had spread westward across the ancient world thousands of years before the New Kingdom mummies were even born. The mummies were also closely related to ancient Anatolian populations and to Neolithic Europeans in ways that modern Egyptians simply are not. That last point is the one that deserves to sit for a moment. The mummies were more closely related to ancient Europeans and Anatolians than to the people living in Egypt today. Modern Egyptians carry approximately 8% more sub-Saharan African ancestry than the ancient mummies. The researchers identified this as a later influx, probably occurring around 700 years ago, well after the period the mummies represented. Something changed in the genetic composition of Egypt's population during the medieval period. The ancient population and the modern population are not the same population. The Egypt that built the pyramids, that produced Ramesses and Akhenaten and Tutankhamun, was genetically distinct from the Egypt that exists today. That is not a political statement. It is what the data shows. The Levant connection did not appear from nowhere. Understanding why ancient Egyptians were genetically closest to ancient Levantine farmers requires going back further than the mummies themselves, much further. To a period before Egypt was Egypt, before the pharaohs, before the dynasties, before the first unified kingdom emerged along the Nile around 3100 BC. Back to the people who were already living in the region when all of that began. The Chalcolithic period, sometimes called the Copper Age, sits between the Stone Age and the Bronze Age. In the Levant, this period produced something remarkable that archaeology had documented but genetics had never fully explained. A specific population, coherent and culturally distinct, occupying the region in a way that left an enormous archaeological footprint but whose origins remained unclear until recently. In 2017, the same year as the Egyptian mummy study, an international team of researchers mapped the genomes of bones recovered from a massive burial complex near Tiye in northern Israel. 22 individuals from a necropolis containing the remains of roughly 600 people. What they found was a genetic signature unlike anything seen in earlier or later populations from the same region. These people sourced approximately 57% of their ancestry from local Levantine Neolithic groups. 26% came from Anatolian Neolithic populations. 17% traced back to Chalcolithic groups from Iran. The combination was distinct. It had not been there before. It would not persist unchanged afterward. And it carried something that the earlier Levantine populations had not shown. Blue eyes, fair skin, genetic markers for light pigmentation turning up in skeletal remains from a region not typically associated with those traits in the popular imagination. The community at Tiye carried recessive genetic characteristics that surprised the researchers precisely because they were unexpected in this geographic context. This population did not persist. The specific genetic combination documented at Tiye disappears from the record. But the ancestry it represented did not vanish entirely. It dispersed. It moved. It contributed to the genetic makeup of populations across a wide region during a period when human movement through the ancient Near East was substantial and sustained. The connection to ancient Egypt becomes clearer when you map the Levantine ancestry in the mummy dataset against what is now known about Chalcolithic population movement. The ancient Egyptians were not simply related to whoever happened to be living in the Levant at one fixed moment. They were related to the deeper ancestral substrate of that region, the accumulated genetic heritage of populations that had been moving through and settling across the ancient Near East for thousands of years before the dynastic period began. What this means for the question of ancient Egyptian identity is not simple. It does not reduce to a single origin point or a single migration event. Population histories never do. But it does establish something clearly. The people who built ancient Egypt carried ancestry that connected them more closely to the ancient farmers of the Levant, to Anatolian Neolithic populations, and to the broader genetic heritage of the ancient Near East than to any population from sub-Saharan Africa. The geography of ancient Egyptian origins pointed northeast, not south. The DNA confirmed it. Egypt is not an isolated case. That is the part that changes everything. The instinct when confronted with a finding like the ancient Egyptian genome data is to treat it as a specific story about a specific place, to file it under Egypt, update the relevant chapter, and move on with a slightly revised version of the history everyone already thought they knew. That instinct is wrong. Because the pattern the Egyptian data belongs to is not regional, it is global, and it is showing up on every continent where ancient DNA can reach far enough back into the record. In 2003, a rock shelter in southwestern Libya yielded 2 women wrapped in woven mats, dead for 7,000 years, preserved by geological accident in a natural desiccation chamber that the surrounding desert should have made impossible. It took 20 years and 3 separate research teams to extract anything usable. When a full genomic analysis finally published in April 2025, the results followed the same logic as the Egyptian data, then went considerably further. The 2 women carried no sub-Saharan African ancestry whatsoever. None. In a region the textbook model had placed at the center of a genetic bridge between North and South, these 2 women had no detectable connection to populations below the Sahara. Instead, they belonged to a lineage that had never been documented anywhere. Not in any living person, not in any ancient genome previously recovered from anywhere on Earth. A ghost population isolated in North Africa for 50,000 years, culturally engaged with the world around them and genetically sealed off from it simultaneously. The Green Sahara had been real. The lakes and rivers and grasslands had been real. Cultural exchange had demonstrably happened. And the populations on either side had not mixed. Culture traveled. Genes did not. The Americas told a version of the same story at a different scale. In 2016, geneticist Bastian Lamas sequenced mitochondrial genomes from 92 pre-Columbian mummies and skeletons collected across South America and Mexico. Remains ranging from 500 to 8,600 years old. His team identified 84 distinct maternal lineages representing the full genetic diversity of the ancient Americas before European contact. Not one of those lineages exists in any living person today. 84 separate lines, all extinct. The diversity that had accumulated over tens of thousands of years was not reduced by European contact. It was erased. Then in 2025 came the first ancient genomes ever recovered from Colombia. 21 individuals from the Bogotá Altiplano. 7 of the oldest belonged to a population documented nowhere else. Their DNA matched no known ancient group in the Americas and no modern population anywhere on Earth. By 2,000 years ago, they had vanished entirely, replaced by a genetically distinct group. A complete population exchange, leaving no descendants. 3 continents. Different time scales, different causes. The same result. Populations alive for millennium, coherent and culturally distinct, gone without leaving a genetic trace in the people who came after them. The ancient Egyptians are part of this pattern, a population whose genetic identity has no clean match in the present, whose closest living relatives are distant, diluted, transformed by centuries of subsequent movement and mixture. The survivors shaped the story. The lost populations did not get to. The evidence deserves honest handling. That means saying clearly what the data shows and equally clearly what it does not. The 2017 Nature Communications study was rigorous. The methodology was sound. The Max Planck team applied contamination screening sophisticated enough to distinguish genuine ancient signal from modern interference with a reliability that earlier approaches could not match. The results held up to peer review in one of the most respected scientific journals in the world. None of that is in dispute. But rigorous methodology applied to a limited data set is still a limited data set, and the limitations here are real enough to matter. 90 mummies sounds substantial. In ancient genomics terms, it is a reasonable sample, but all 90 came from a single site. Abusir el-Malek was one town in one region of a country that stretched for 1,000 kilometers along the Nile. Whether the genetic profile documented there accurately represents the full population diversity of ancient Egypt across its entire geographic and temporal range is a question the data itself cannot answer. Egypt was not one community. It was a civilization spanning millennia and thousands of kilometers of territory. 2 or 3 full nuclear genomes from one location in one period is a beginning. It is not a conclusion. The geographic objection extends further. Upper Egypt and Lower Egypt had distinct histories, distinct population pressures, and distinct contact patterns with surrounding regions. The Delta region in the north faced the Mediterranean world. The southern reaches bordered Nubia. A population sample from a site near modern Cairo tells you something specific about that location. It does not automatically speak for Thebes or Memphis or Elephantine or any of the dozens of other centers of ancient Egyptian civilization. There is also the question of time depth. The mummies at Abusir el-Malek spanned from the New Kingdom through the Roman period. That is 1,400 years of history. But ancient Egypt, as a coherent civilization, stretched back another 2,000 years before the earliest mummy in the dataset. The pre-dynastic period, the early dynastic period, the Old Kingdom that built the pyramids at Giza, none of that is represented in the genetic record yet. Whether the population that built the Great Pyramid carried the same genetic signature as the New Kingdom mummies at Abusir el-Malek is currently unknown. The modern comparison databases carry their own limitations. Ancient sequences are compared against present-day populations to identify the closest living relatives. But those databases are not complete. Sampling of modern populations across North Africa, the Levant, and the broader ancient Near East remains uneven. Populations that no longer exist as distinct communities may have carried genetic signatures that would change the picture if they were included. Absence of a match in a database is not the same as absence of a match in the world. What is not in dispute is the core finding. The ancient Egyptians were not genetically continuous with modern Egyptians. Something changed. The population that exists along the Nile today carries significantly more sub-Saharan African ancestry than the mummies do. That shift is real, and it happened after the period the mummies represent. What caused it, how fast it happened, and what it meant for the people living through it remains an open question. The data opened the door. It did not walk through it. Return to the mummies in the Egyptian Museum. They were not symbols of a debate. They were people, farmers, administrators, queens, rulers, members of a civilization that lasted longer than most of human history can comfortably hold in a single thought. They did not know their DNA would one day contradict everything the modern world assumed about them. They did not know they would become evidence in an argument that had not yet begun. The science that found them is not finished. It is barely started. The Old Kingdom remains unsampled. The pre-dynastic period is almost entirely dark. There are entire centuries of Egyptian history where the genetic record simply does not yet exist. Every blank space is now a potential ghost. The mummies have been waiting 3,000 years. The questions the DNA is forcing are not going to resolve quickly, but they are the right questions. We are only beginning to learn how to ask them.